Hey, how are we doing everyone? Welcome to the Bearded Medic. So in the last video we went through the innate response to an extracellular pathogen. What we're going to do now is go through the innate response to an intracellular pathogen, including how cells sample themselves to show if they're infected or not, and how natural killer cells are involved in the innate response. So we have our DAMPs from cell damage and PAMPs, which are parts of the pathogen, which must be identified by pattern recognition receptors. For extracellular pathogens, as we mentioned, they are phagocytosed upon recognition by phagocytic receptors. So let's take our bacteria again that's been opsonized by complement. It's recognized by complement receptors and internalized in the process of phagocytosis. But some bacteria can survive the harsh environment of the phagolysosome and escape into the cytoplasm of the phagocyte. But the body has a defense against this: intracellular pattern recognition receptors. These are certain toll-like receptors and nod receptors for bacteria, and other toll-like receptors and rig-like receptors for viruses. If we take intracellular bacteria, the peptidoglycan cell wall of bacteria is recognized by nod-like receptors. This initiates the same response we went through in the last video. If you remember, there's increased activation of the NF-kappa B transcription factor, release of pro-inflammatory cytokines and chemokines, which lead to inflammation to recruit immune cells to destroy as much pathogen as possible. But what if we have a pathogen like a virus? Replicating inside the cell, it's less likely that a tissue-resident immune cell will come across a pathogen outside the cell, and our nod receptors only recognize the peptidoglycan cell wall. So we need certain intracellular toll-like receptors and rig-like receptors to identify components of viruses. Recognition by these receptors leads to the increased activation of transcription factor interferon regulatory factor, or IRF for short. And to make it more complicated, just as there's multiple TLR subtypes, there are also variations in the IRF transcription factor. Recognition by TLR3 and RIG receptors leads to IRF3. Recognition by TLR7, 8 and 9 leads to IRF7. This depends on what component of the virus is being recognized. But for now, know that IRF leads to the release of class 1 interferons. Interferons are cytokines. And what are cytokines? Signaling molecules. The class 1 interferons are called interferon alpha and beta. They enhance both the innate and adaptive response, but let's go through their actions in the innate response. On the adjacent cells to the infected cell, they bind onto interferon receptors. They essentially warn themselves that there's a virus, telling them to shut down any mechanisms that can aid viruses. But how do they do that? So they go to adjacent cells and activate what's called the JAK-STAT pathway. This leads to a bunch of signaling cascades, leading to new genes being activated. These genes increase cell RNase L, which degrades viral RNA, so the viruses can't replicate. Phosphorylate proteins involved in translation, which blocks viral protein synthesis, so the viruses can't synthesize new proteins. They inhibit virus assembly, so new viruses can't be fully formed, and increase the major histocompatibility complex 1, or MHC1 expression in cells, essentially meaning cells sample their inner contents more often, meaning virally infected cells are more likely to be picked up by the immune system, namely natural killer cells. Before we get to the role of natural killer cells, let's look at the MHC molecules because these are key. There are two types, MHC1 and MHC2. MHC1 is expressed on all nucleated cells, but MHC2 is only expressed by antigen-presenting cells, which are macrophages, B cells, and dendritic cells. MHC2 processing will be covered in another video, but let's explore MHC1. So when cells sample their own inner contents, they display it on the molecule MHC1. Cytosol proteins are degraded by a proteasome into small peptides. These pass on to the endoplasmic reticulum of cells and are packaged on the MHC1 molecule, which transports them to the cell surface membrane to be displayed to external cells. So why do they do this? To prove they are healthy cells. Now imagine a virus had colonized the cell. Parts of it may have been processed by the proteasome and transported on MHC1 to the surface, and that shows the cell has been infected. Now this is where natural killer cells come in. They're like sentinels of the immune system. Any sign of trouble in a cell equals elimination. They have two types of receptor. One is an activating receptor and one is inhibitory. The activating receptor binds to certain ligands. The natural instinct of these cells is to kill, hence the name, so these activating receptors initiate this instinct. However, the inhibitory receptor responds to MHC1 and the peptides displayed on it. If the peptide on MHC1 is self, then the inhibitory receptor is stimulated. Why? The cell is healthy, it poses no threat as a vessel for making more pathogen, so there's no need to kill it. So the inhibitory receptor suppresses signals from the activating receptor, meaning cell death is not initiated. But if the peptide is foreign, the inhibitory receptor notices that the presented peptide is not self. So there's no inhibitory signal, the activating signal predominates, and the natural killer cell initiates cell death. Now how does it do this? Now if we look at these circles, they have perforins, and as the name suggests, they perforate or puncture the cells. They also contain granzymes. They are secreted into the cell, which triggers apoptosis, which is cell death, leading to destruction of the infected cell. But viruses also have a neat trick where they can decrease MHC1 expression from cells. You would think that means they're less likely to be detected, but the inhibitory receptor also detects there's no peptides on MHC1. And this is a problem to the natural killer cell. The cell has nothing inside it, so the activating receptor predominates and induces cell killing. Also as a mechanism for the cell, when viruses are inside cells, ligands that activate the receptor on the natural killer cells are upregulated, so more activating signals are initiated on natural killer cells if they come across an infected cell. So overall, natural killer cells induce cell death in infected cells. They are specific to only those cells that have been infected, sentinels that snuff out any sign of danger. They also have a couple of other functions to note. If you remember phagocytosis, macrophages that phagocytose not only release the pro-inflammatory cytokines, but they also release the cytokine IL-12. This attracts natural killer cells to macrophages. They interact by sending signaling molecules to each other. The main ones to note is interferon gamma that is released by the natural killer cells to the macrophages. This changes the macrophage phenotype or action, making them more aggressive to pathogens. So essentially you get more phagocytosis and more destruction of pathogen. Interferon gamma is a class 2 interferon. So let's review the concept of intracellular recognition of pathogens. Intracellular pattern recognition receptors identify the pathogen as foreign. Depending on the pathogen and the receptor, they react accordingly. Recognition of the bacterial cell wall by nod-like receptors leads to the NF-kappa B pathway, which leads to the release of pro-inflammatory cytokines and immune cell recruitment, like the extracellular pathway. Recognition by toll-like receptors and Rig-like receptors leads to the ERF pathway, producing type 1 interferons. They warn adjacent cells about the pathogen and upregulate antiviral mechanisms in those cells to decrease viral protein synthesis, and get them to display more of their inner contents on the MHC1 molecule. Natural killer cells can identify if a cell is infected if non-self peptides are displayed on MHC1. They have an activating receptor which is stimulated to induce apoptosis, and an inhibitory receptor which does the opposite and allows the cell to survive. If the cell is infected, the inhibitory receptor is less stimulated, meaning the activating receptor signals predominate, which leads to cell death by perforin and granzymes. This whole process means natural killer cells aim to kill as many infected cells as possible. So hopefully you can tell the principles of recognition remains the same no matter where the receptor is and what the pathogen is. A pattern recognition receptor identifies the pathogen, a signaling cascade increases the activation of a specific transcription factor. This codes for certain cytokines, which are signaling molecules that enhance the response according to the pathogen that has been encountered. All this within a time span of 12 hours. The hope is to destroy that pathogen then and there, which works most of the time, but what if it doesn't work, or we want to remember that pathogen for next time? We need a more specific response to the pathogen, and that is the adaptive response, which we will begin with in the next video. Thank you for watching. Hope you found the video helpful. The best way to make it known is by leaving a like. Be sure to comment if you have any questions and come over to Instagram to test yourself. See you in the next video. Goodbye.